So good evening, everybody. Thanks all for be patient. Thanks for the invitation. As you guys mentioned, I'm Christos Papadelis. I'm the founding director of neuroscience research at Cook Children's Healthcare System. And I'm also professor of research in bioengineering at the University of Texas at Arlington and professor of pediatrics in Texas Christian University. Today I will speak about the neuroimaging correlates of GABA metabolism in children with Dravet syndrome. And I would like to say that I'm a very new researcher in Dravet syndrome, despite the fact I have worked in epilepsy for a long time. These are my financial disclosures, and upon completion of my talk, you will be able to identify the pathophysiological mechanisms of GABA dysfunction in children with Dravet syndrome and other GABA metabolic disorders. Identify the neurophysiological biomarkers of neurotransmitter metabolism using electroencephalography or EEG and magnetoencephalography or MEG, and understand the link between GABA metabolism and high-frequency neural activity in the human brain. I know that this is basic knowledge, but I will just repeat that. So we know that Dravet syndrome is a rare epilepsy disorder, affects approximately 1 to 15 with 1 to 40,000 live births. The patients, they usually suffer with seizures, with an onset at 5 to 8 months of age. And these kind of seizures at the beginning, they're usually triggered by fever. And within weeks to months, they also develop additional febrile seizures and also temperature-independent seizures, which are severe and disabling. And as it was mentioned before, Dravet syndrome, patients, they don't suffer only from epilepsy and seizures, they have also several other comorbidities. And the first mutation that was linked with Dravet syndrome was identified back in 2001 in the SCN1A gene that encodes the A1 subunit of the voltage-gated sodium channels. And why the voltage-gated channels are important? They play a critical role in the initiation of axial potentials in the neurons and in the regulation of their excitability. You can imagine, for the families, with that axial potentials, actually electrical signals that they travel from neuron to neuron within the brain, and they transfer all this kind of information. The sodium channels, they are located at the axial hillocks and across the length of the neural axons. And during the resting period, when the neuron is in a resting condition, the channels are initially closed, so there's no any kind of influx or outflux of ions across the membrane. And when we have a nerve pulse that arrives, the sodium channels, they are activated. They open the gate and allowing positively charged sodium ions to enter the cell. So following activation, the channel does not open in response to a new signal for a brief period of time. And this critical sequence of events dictate the propagation of axial potentials across one specific direction to the neurons. Human genetic studies and recent animal work suggest that precise control of ion flux, such as the one from the voltage gate sodium channels, contribute significantly to the developmental process. So several inherited or sporadic gene mutations that encodes brain ion channels will affect the normal human brain development and cause neurodevelopmental disorders. Most of these disorders, they are characterized by epilepsy, as well as cognitive and behavioral deficits and significant phenotypic and genetic heterogeneity. Studies on patients with Dravet syndrome have shown that mutations in the SCN1A gene typically produce non-functional channels, and these kind of non-functional channels will cause reduced sodium current, and that's less sodium depolarization and inexcitability in the neurons. So the main idea is that the Dravet syndrome mainly arises from what's called de novo in genetics. It means occurs for the first time in this family. SCN1A haploinsufficiency. It's a kind of a genetic term that indicates in situations which the total level of a gene product produced by the cell is about half of the normal level, and that is not sufficient to permit the cell to function normally. However, there's a kind of a paradox here. It's a surprise that reduced expression of a gene that encodes an essential depolarization current like cause epilepsy. And this is because like reduced sodium current should lead to inexcitability rather than hyperexcitability. And in order to explain this kind of paradox, and we saw some other previous speakers like they touched about that, there have been alternative hypotheses that have been proposed, mostly based on findings in animal studies. And the main one is like what's called the Interneuron Hypothesis. So patch clamp recordings in animal models, they have shown that like bipolar GABAergic neurons, like they have a kind of a dramatic reduction in sodium current density. And this loss of sodium current density from the GABAergic neurons like limits their ability to increase like firing frequency in response to injected current. So based on these results, it was hypothesized that like seizures in Dravet syndrome, they arise as a kind of a result of selective defects in the GABAergic interneurons, which produce a network that lacks sufficient inhibitory tone. So you can think of this kind of GABA deficit as a kind of a car in which like the brakes, they don't work properly or they are broken, or an electrical circuit in which like the off switch does not work. And let's talk a little bit about the Gabaergic system. So the Gabaergic system is one of the two main important amino acid systems in the brain. The glutamate system is responsible for fast signaling excitation. The GABA system is responsible for fast signaling inhibition. So it's a kind of an off switch in a large electrical circuit that responds very quickly and has like a kind of a global effect in the brain, since nearly half of all synapses in the brain, they express some kind of GABA receptor. We also know now that GABA plays a significant role not only in humans, but in almost like all species. It's, nearly all organisms ranging from the bacteria to the humans, they can synthesize GABA. The GABA neurotransmitters act at the synaptic level by modulating ion channel activity to two major postsynaptic receptors, the GABA-A ionotropic receptor and the GABA-B metabotropic receptors. So upon GABA transmission binding, the GABA-A receptors, they open, allowing negatively charged chloride ions like to flow into the neuron, make it like more negative or more hyperpolarized, and that's less likely to generate axial potentials. Upon GABA transmission binding, the GABA-B receptor is also activated and opens and allows like positively charged potassium ions to flow out from the cell, making the postsynaptic neuron like hyperpolarized, and that's less likely to generate any kind of axial potentials. But the gamma neurotransmitter did not always like have kind of an inhibitory role. We know now that at the earliest stages of the development, GABA produced an excitatory response instead of an inhibitory response as seen in the mature brain. So the direction of the GABA current changed like, and actually reversed during brain development from depolarizing to hyperpolarizing. And this kind of developmental change is referred to as postnatal GABA shift. It plays a critical role in the human brain development. It was mentioned before that the proper functioning of the central nervous system like depends on the balance between excitatory and inhibitory neurotransmitter systems. The excitatory system is regulated by glutamate. The inhibitory system is regulated by GABA through interneurons, which they modulate the excitatory level generated by glutamate release. So these interneurons actually, they control the information flow and the synchronization of the cerebral cortex, and can be seen in several areas of the brain, such as the amygdala, the hippocampus, and the prefrontal cortex. This wide expression of the GABAergic cells indicate that this kind of inhibitory neurotransmitter is involved like in many functions in the central nervous system, such as the cognition and the motor control. But the problem is like GABA cannot penetrate the blood-brain barrier, and that's its measures through blood tests do not directly reflect the levels of the GABA in the brain. So there's a kind of a recent interest to develop non-invasive tools, which are based on neuroimaging methods that can provide a kind of an indirect measure of the levels of the GABA in the brain. There are several neuroimaging methods which can help us like to assess in an indirect way the levels of the GABA in the brain. So among them today we'll talk about like MEG or magnetocephalography, the high-density EEG or high-density electroencephalography, and the magnetic resonance spectroscopy, or MRS. The MEG is a non-invasive neuroimaging technique that measures the magnetic activity of the human brain. It actually presents like several advantages compared to other neuroimaging methods, particularly for kids. The patient sits comfortably in an armchair and placing his head inside of a helmet that contains like several sensors. These sensors, they are called gradiometers or magnetometers. And the MEG measurements, they are based on the phenomenon of electromagnetic induction, according to which like any kind of changing magnetic field induce in a nearby coil an electrical current. So we have like several thousand of neurons in a focal area of the brain which are simultaneously active. These neurons, they generate an electrical current. The electrical current generates a magnetic field, and what we measure outside of the scalp through this kind of coils is this kind of weak brain magnetic activity. A technique that is comparable to the MEG is the high-density EEG that measures now the electrical activity of the brain. Modern high-density EEG systems allow the recording of up to 256 electrodes covering the entire patient's head without even the need to apply for any kind of gel. So we have EEG nets, which next to each of these electrodes, there are attached sponges. We just simply soak this kind of nets in a saline solution, and then we place the net to the patient's head, and we have immediately conductivity. This kind of setup is ideal for pediatric patients or patients with challenging behaviors. And we're also now able to combine the MEG and the high-density EEG recordings to a state-of-the-art setup we have here at Cook Children's Medical Center, which allows the simultaneous recording of human brain activity with more than 550 sensors that cover the entire child's head. Magnetic resonance spectroscopy, or MRS, it's another neuroimaging technique that allows to examine the human tissue for the presence and concentration of different metabolites, including the GABA. The MRS spectroscopy is conducted on the same machine as the conventional MRI, and that could be easily integrated with any kind of conventional MRI scan. So while with the conventional MRI, we use the signal from hydrogen protons to form anatomical images like this one you see here, MRS using the information to determine the concentration of brain metabolites. There are three basic neurophysiological mechanisms which are candidates to generate the EEG or the MEG signal that we measure outside of the scalp. We have axial potentials propagations traveling along the axon away from the soma. We have postsynaptic currents at the apical dendrites of the neurons and back-propagating spikes into the dendrites. Among these kind of three mechanisms, the postsynaptic currents are believed to be the ones that they are measured by the MEG and the EEG. However, recent evidence from my group and several other groups, they have shown that axion potentials may also contribute to what's called high-frequency oscillatory activity, which can be measured with the EEG and the MEG. A first step towards tools that allow us to measure levels of brain GABA non-invasively, it is the observation that drugs such as the benzodiazepines, which potentiate the GABA-induced chloride influx, affect the oscillatory patterns of the EEG signal. And although benzodiazepines, they are sedative drugs, so we expect a kind of suppression of the EEG activity, recent animal studies have shown that they actually increase the beta and gamma activity in the EEG signal, and possibly this is through their action on the GABA-A receptors. The question is now how we can induce this kind of frequency-specific changes in the human brain and how we can measure them with non-invasive tools such as the EEG or the MEG. So early studies on patients with implanted intracranial EEG have shown task-specific high-frequency activity between 40 and 150 Hz in the raw signal in response to visual stimuli presented to the patient. By using more advanced neuroimaging tools, such as the MEG, recent studies have shown gamma band activity in frequencies between around 60 Hz, which were induced by the presentation of visual stimuli in healthy adult subjects. And by also using MEG, another group of researchers showed evidence of gamma-induced activity in the frequencies between 60 and 95 hertz in the primary somatosensory cortex of healthy adults in response to painful stimuli. However, in order to measure this kind of activity non-invasively, we need also modern neuroscience experimental designs. And here we see, for example, a visual experimental setup in the MEG at Cook Children's Medical Center. So we actually present to the child like several visual stimuli in a screen that is located in front of the child, and we measure the brain activity in response to this kind of stimuli. For the enhancement of the gamma-induced activity, we present like flashing checkboards that is well known that generates the strongest possible cortical response within the primary visual cortex. In order to assess this kind of weak brain responses, which can be easily recorded with implanted electrodes, we need also to increase what is called the signal-to-noise ratio, or the quality of the EEG and the MEG signal that we record non-invasively. And in order to do so, we adopt a methodology that is based on the electric and magnetic source imaging. It's a kind of two mathematical complex problems that I will not go into details, the forward problem and the inverse problem. What we do, we initially co-register the location of the EEG and the MEG electrodes with the patient's head scalp extracted from its MRI. And based on this information, we develop a kind of a head model that approximates the patient's head electromagnetic properties. By using this model, we're able to estimate what we call virtual channels within predefined locations in the patient's brain non-invasively. And this kind of virtual channels allow us to have a very high signal-to-noise ratio brain activity, which can be recorded non-invasively instead of implanting electrodes inside of the brain. By using these two kind of methodological approaches, like my team has shown in research studies that we can detect indeed non-invasively what's called high-frequency activity between 80 and 250 hertz, and this is called ripples with the scalp EEG and the MEG. And this kind of activity was often overlaid on indirectal spikes and can be seen only in patients with epilepsy. In healthy adults, a MEG group from UK showed for the first time evidence that the amplitude and the frequency of gamma-induced responses to visually presented stimuli that was recorded with a MEG was positively correlated to the resting state GABA brain levels assessed with magnetic resonance spectroscopy. So they actually performed a similar experiment with functional MRI and found that bold amplitude of the hemodynamic response in the primary visual cortex was negatively correlated with the GABA level and the gamma frequency. Another research group from CHOP in Philadelphia showed similar findings in the primary motor cortex with the MEG. By using the virtual channel approach that I described before, they found what is called movement-related gamma synchrony in the frequency of 80 hertz in response to finger tapping movements in a virtual sensor that was located in the contralateral primary motor cortex to the active finger. This kind of activity was preceding what is the well-known event-related beta desynchronization and the post-movement beta rebound, these kind of activities that we observe in the brain when we move our fingers. And the amplitude and the frequency of this gamma activity was positively correlated with the levels of the GABA, as this was measured with magnetic resonance spectroscopy on the same subjects. More recently, a group from Germany examined the correlation of the GABA levels with the gamma high-frequency induced response in the auditory system. By using magnetic resonance spectroscopy, they actually estimated the level of the GABA in healthy individuals on a large voxel covering the primary auditory cortex. By using EEG and the passive listening task, they recorded brain responses to auditory stimuli from healthy adults. And although they managed to successfully record gamma-induced activity that was positively correlated with sound intensity, they did not manage to reproduce the findings from the previous studies. A few studies now have emerged the last few years trying to correlate the levels of brain GABA with EEG biomarkers in neurological disorders where the Gabaergic system is involved. And here we see the findings of a resting state EEG study on patients with DUP15Q syndrome. Is the most common form of genetic cause of autism spectrum disorder, and a group of healthy controls. So what actually they observed a significant increase in the EEG power within the beta frequency band compared to the healthy controls. In a group of adults with a diagnosis of schizophrenia, it was found a relationship between in vivo brain GABA levels measured with magnetic resonance spectroscopy in the median prefrontal cortex and auditory inhibitory electrophysiological measures with EEG. So by using a per-click gating paradigm, they actually observed a negative correlation of gating for theta-alpha ratio and beta activities with the levels of the GABA measured by magnetic resonance spectroscopy. There's no many studies on Dravet, unfortunately, and that's why it's a very interesting topic for me, because I have done all this work in other disorders, but Dravet is something new, and then this is the only study on Dravet that they have used EEG so far. It's a study from Europe that tested the hypothesis that brain oscillator activity in the gamma frequency band will be altered as a result of GABA dysregulation in the brain of patients with Dravet syndrome. So actually they examined with EEG 13 children with Dravet syndrome using an auditory state-state experiment and compared the EEG findings with a group of children with non-Dravet epilepsy and a group of healthy controls. The experiment was involving the delivery of auditory stimuli that was provided to the subjects through earphones in the frequency of 1,200 Hertz that was modulated in amplitude. Here we see the time frequency representations of the auditory evoke responses from a single participant from each group, and we see that like for the child with Dravet syndrome, there was no any kind of activity, or this activity was suppressed compared to the other two groups. So this European group observed weaker or no responses in the 1 to 120 Hertz frequency range for patients with Dravet syndrome compared to both healthy controls and children with non-Dravet epilepsy. And actually healthy controls, they showed oscillatory responses that they were maximum in the low and the high gamma ranges, both in the power and the intertrial coherence estimates. So these kind of findings indicate that suppression of gamma activity in children with Dravet syndrome compared to healthy controls, that supporting our hypothesis of GABA suppression in children with Dravet syndrome with a non-invasive biomarker. As I said before, Dravet is a new syndrome for me, but I've done work on another Gabaergic disorder called SSA-DHD. It's an ultra-rare genetic epilepsy disorder that has several similarities with Dravet syndrome, and it's also possibly caused by GABA deficiency. This work was performed in my previous institute at Boston Children's Hospital together with the PI, Philip Pearl. And we hypothesized that these patients with SSA-DHD will present altered somatosensory brain responses in the beta and the high gamma frequency band compared to healthy controls. So in order to test our hypothesis, our team recorded somatosensory evoke potentials with high density EEG from 17 patients with SSA-DH deficiency and 10 healthy controls. And in order to elicit these somatosensory responses, we deliver to the subject's fingers pneumatic stimuli through compressed air puffs. By solving the inverse problem that I described before, we estimate this kind of virtual channels, and we extracted from these virtual channels the brain activity non-invasively. We also assessed for the same participants the level of the GABA in the patient's blood through plasma specimens and in their brain through magnetic resonance spectroscopy. This is unpublished data, so we have a paper that we are preparing right now to submit. And here we see what's called the time frequency analysis plots among all participants for the two groups. This is the SSA-DHD group, or Gabaergic deficit group, and the healthy control group. What you see with red color is higher activity in the brain in response to this somatosensory stimuli. And what we observe a significant suppression in the beta and lower and gamma frequency band for the SSA-DHD group compared to the healthy controls. We observe a significant increase in the GABA level in the blood samples of children with SSA-DHD compared to healthy controls. But in contrast, we observe a significant decrease in the beta and the gamma power at 3 specific time windows for the patients with SSA-DHD compared to the controls. Finally, we observe a moderate positive correlation between the participants' age and the suppression of the beta activity. This is the correlation findings. So we correlate the different findings from the blood and the brain responses. So we observed a very strong negative correlation between what's called event-related powered synchronization in the beta frequency band and the levels of the GABA in the blood among all participants. And from this graph, we can see a clear classification between the patients with SSA-ADSD and the healthy controls in terms of like beta synchronization and the levels of the GABA in the blood specimens. We observed similar findings for the gamma activity at the early time window, a moderate correlation between event-related power synchronization in the gamma frequency band and the levels of the GABA in the blood among all participants. Based on all this evidence I presented today, we have initiated a project here at Cook Children's which Encoded Therapeutics have agreed to provide funding, having as a main goal to develop non-invasive tools that assess the levels of the GABA in the brain and correlate these kind of findings with the levels of GABA in the blood. So in this study, we plan to use a combination of different neuroimaging techniques and correlate these kind of indirect measures of GABA with the ones derived from measures of the blood. These kind of efforts can increase our understanding of the pathophysiology of Dravet syndrome, as well as other neurological disorders of GABA metabolism, and validate non-invasive systemic biomarkers of brain GABA metabolism that can serve as essential monitoring tools in future clinical trials for patients with Dravet syndrome. I would like to thank my group for all the help and support, but most importantly, I would like to thank the patients and their families who participate in our studies. Thank you.
Do we have any questions?
Thank you, Christos. Great talk. Quick question. Did you also do MRS in the SSA ADHD? And if you did you compare GABA levels against the GABA levels in blood?
Yes, we did. I just didn't present this data here. Okay. And then we have an interesting correlation. The problem with MRS, it's a kind of a quite challenging technique because in order to extract this kind of activity from a large voxel, the child should stay still for a relatively large period of time. And kids with SSA-ADSD, they were they were able to stay still for some time, but not all of them. So it's a kind of challenging technique to propose in challenging populations. If, for example, a child that is not able to still for some time, then you should repeat the recording and then again and again. So it's a kind of a hassle. But the findings are interesting and important. Yeah.
Thank you.
Do we have any other questions? All right, thank you so much.
Thank you.